Hi there, thank you for taking the time to listen to this research presentation. I want to go through some data with you today, which were presented at the 132nd annual meeting of the Indiana Academy of Sciences. The title for the presentation is Identifying Gene Expression Changes that Reduce Age-Related Bone Loss. This work was performed in collaboration with two medical students, Michael Eaton, who is currently a third-year medical student at Marian University, and Aaron Hudnell, who is currently a fourth-year student at Marian University. For further information about my research program, you can follow the link here, www.johnlowery.com/research. You may follow the lab on Twitter at #LoweryLab. My Twitter handle is @JW_Lowery, where you can find information about the Bone and Mineral Research Group at Marian University, of which I'm the director. My students are active participants at the Twitter handle @BMResGroup. There are no conflicts of interest to disclose for this presentation. My lab is very interested in understanding the mechanisms that control age-related bone loss in humans. We frequently use mouse models of age-related bone loss or changes in bone development to understand the cellular and molecular changes that might be future targets for addressing age-related bone loss in humans. As you may be aware, bone loss begins in the third or fourth decade of life in humans. What is shown here on the panel are data from 2010 by Menelagus, representing changes in femoral neck bone mineral density in women of three different ethnicities. And you'll see that in the third or fourth decade of life, bone mass begins declining, and this trend continues throughout the remainder of the individual's life. Now, all ethnicities that are described in this panel experience age-related decline, and what you'll notice is that the major determinant of who reaches quote, low bone mass first is largely determined by peak bone mass as young adults. The data that are shown here, as I mentioned, are for women, but correlating changes are also observed in the skeletons of men. At the architectural level, it may help to conceptualize the changes that are resulting from age-related bone loss using these images here, which would be high-resolution micro-CT images of normal bone mass. This would be a type of bone called trabecular bone. This is the type of bone also called spongy or cancellous bone that has these trabecular structures, you can imagine like struts, which are interconnected with one another, providing compressive strength to the bone. In conditions of low bone mass, such as osteoporosis, which is a diagnostic kind of criteria quantifying the change in an individual's bone mass, to be greater than or equal to 2.5 standard deviations away from mean bone mass for gender and ethnicity matched individuals. In osteoporosis, there's a thinning and progressive loss of the connections between these trabecular structures of cancellous bone. You'll notice that the spaces here between the struts are wider and the struts themselves are thinner. Understanding the mechanisms behind age-related bone loss in humans is on some level a personal mission of mine. The women in my family, many of them are affected by osteoporotic-related bone loss. This is a picture of my grandmother. Her name was Myrtle McKay. She passed away a few years ago after having suffered fractures to both hips and one wrist due to osteoporosis. Now, osteoporosis did not cause her death, but it certainly contributed to the comorbidities she experienced later in life. And in fact, up to 25% of hip fracture patients die within 1 year of their injury, and some reports have the 30-day mortality as high as 10%. There is, though, a very large treatment gap among the patients with osteoporosis in the U.S. In fact, the treatment gap is about 80%, where only 1 in 5 patients over the age of 50 who suffer a hip fracture go on to osteoporosis pharmacotherapy. My grandmother was never treated for osteoporosis. Now, the incidence of osteoporosis is expected to rise as the worldwide population ages. In men, the expected increase is over 300%, and in women, it's nearly 250%. This is by the year 2050. In Indiana alone, the projections indicate that by the year 2020, 1.3 million Hoosiers will be at risk for developing osteoporosis or already have osteoporosis based on strict diagnostic criteria. The mechanisms leading to osteoporosis are clearly complex. There are numerous targets that have been reported, and there are several lines of investigation that would suggest different molecules that may be used to reverse age-related bone loss. No matter what the mechanism is, it's helpful to think about the changes in bone mass that occur with aging in terms of a seesaw. Many of us experience playing on seesaws on playgrounds, where I've seen children playing on such structures. And the idea is that there are two opposite weights that are resting on top of a fulcrum. And the relative weight of the left side versus the right side impacts the balance across this fulcrum. In general, it's agreed upon that in the aging skeleton, bone resorption, which is carried out by a type of cells called osteoclasts, outpaces bone formation, carried out by a type of cells called osteoblasts. The net effect of this is bone loss. Current frontline strategies, such as bisphosphonates or a monoclonal antibody called denosumab, are considered anti-resorptive therapies. These are the first-line approach where an attempt is made to rebalance the seesaw by reducing the amount of bone resorption that is occurring by inhibiting osteoclast function or causing apoptosis, loss of osteoclasts. Now, anti-resorptive therapies can be very helpful at maintaining bone mass, but some individuals have already staggeringly low bone mass or are at extremely high risk of fractures. For those individuals, the recommendations are for anabolic therapies. At the moment in the US, there's only one FDA-approved anabolic therapy, while there are two other additional anabolic therapies that are in the pipeline. The one that is approved is called teriparatide, that is produced by Lilly and Company as the trade name Forteo. One of the other molecules in this pipeline is called abaloparatide. The mechanism of action is similar to teriparatide. And then the other anabolic therapy that is in the works is a monoclonal antibody against the sclerostin gene product. In my mind, these things, the situation suggests that we have an urgent and unmet need for developing new strategies that address age-related bone loss. My entrance into the field of osteoporosis was somewhat accidental, where while working as a postdoc at Harvard School of Dental Medicine, I was working on a mouse model that involved deleting the BMPR2 gene from the appendicular skeleton of mice during embryonic development. For the technical details, you can follow the link here to this article. The study began by looking at bone development, but we immediately noticed that these mice didn't have a problem in bone development per se, but the loss of BMPR2 led to high bone mass as young adults due to an increase in osteoblast activity. This work was performed in collaboration with numerous individuals, and the senior author for this paper is Vicki Rosen, who was my postdoc advisor at Harvard School of Dental Medicine. I want to show you just a couple of pieces of data from this publication to demonstrate to you how the bone mass phenotype appears in these animals. These are micro-CT images of tibia and fibula from 9-week-old mice. On the left, this would be a wild-type tibia and fibula, and on the right, this would be a BMPR2 mutant tibia and fibula. I would point out to you the cortical shell, which is the bone here on the exterior of on the outsides of the tibia, is thicker as compared to the controls, and also the fibula is almost completely filled by bone as compared to the controls. The other thing to point out is this trabecular bone, which you saw by micro-CT imaging in an earlier slide. Quantitatively, the changes in the BMPR2 mutant mice were revealed to be a greater than 60% increase in volumetric bone mineral density, which would be a parameter that is evaluated in humans, and this was due to an elevated bone formation rate. Now, we never observed a change in bone resorption in these animals. Currently, my lab is following up on these studies via several means. The first is examining what happens to the bone mass in BMPR2 mutant mice with aging. Second, what gene expression changes underlie elevated bone formation rate in BMPR2 mutant mice? And third, can we move away from this embryonic genetic approach to target BMPR2 function of the adult skeleton pharmacologically? And finally, what changes in cell-to-cell communication are associated with high bone mass in BMPR2 mutant mice? We have several pathways of particular interest to our lab, and we are performing signaling studies in these bones now. Now, for the remainder of the talk, I want to focus on these first two questions, which are interrelated. Just like in humans, we observe in our hands that these control mice, which are on the C57 Black 6 background, experience an abrupt decline in bone mass as they age. The circle here on the left represents the mean bone mineral density of a cohort of 15-week-old control mice in the 95% confidence interval to 55 weeks of age. Across this time period, these wild-type mice, control mice, experienced approximately 70% decline in bone mineral density. BMPR2 mutant mice, which are indicated here in the red circle, the mean and 95% confidence intervals again, as we previously reported, have higher bone mass as young adults. And we were interested to observe here that the slope of the decline of bone mass between the mutant mice and control mice differs. In particular, the slope is less steep in BMPR2 mutant mice. Across the same time span, while wild-type mice experience about 70 milligrams of hydroxyapatite loss, BMPR2 mutant mice experience somewhere in the neighborhood of half of that amount of bone loss. We then moved on to look at markers of bone formation activity or bone resorption activity in the serum of these animals. We began by looking at 55-week-old individuals. The wild-type mice are represented here in black circles, whereas the BMPR2 mutant mice are in red circles. We found no difference in bone formation activity as measured by P1 and P levels in the serum at 55 weeks of age. But we wondered, what happens if we back up to an earlier time point? So we moved up this curve to 35 weeks of age. At 35 weeks of age, we were able to observe an increase in bone formation rate as measured by P1 and P activity in BMPR2 mutant mice as compared to control littermates. Like in earlier time points, we have yet to observe a change in bone resorption parameters at these later time points as well. The working hypothesis based on these data is that loss of BMPR2 in the skeleton leads to a sustained increase in osteoblast activity into late adulthood. We applied for a senior research grant to the Indiana Academy of Science to follow up this study by determining the gene expression changes that are associated with increased bone formation rate in BMPR2 mutant mice. The time point we chose was here at 35 weeks of age, which was the latest that we had observed increased bone formation rate in these animals. And we thought by so doing that we'd be able to observe what might be a smoking gun. While BMPR2 mutant mice still have an elevation in P1 and P activity at this time point, control mice are experiencing a significantly lower bone formation rate, and we thought this would be the appropriate time point to exploit those differences. The technique we chose is called RNA-seq. It's also called transcriptome profiling, where we began with 4 wild-type humeri and 4 BMPR2 mutant humeri. We flush the marrow out of these bones by gentle centrifugation, and you can see this would be when the marrow is present, and then these bones here on the right, these 4 on the right, are after the marrow has been removed. The bones were homogenized and total RNA was collected. And then, because we are aware that there's a good deal of technical variability in our isolation technique, we decided to pool these samples together so that we're looking by RNA-seq at an average change for the genotype. We then submitted the pooled RNA samples. This would be 4 pooled control samples and 4 pooled BMPR2 mutant samples to GeneWiz. Quality control was performed there, followed by ribosomal RNA depletion and synthesis of a library, which was then sequenced. Bioinformatic analyses were performed along with differential gene expression, and we moved forward. Now, at this time point, we anticipated finding 3 groups of genes. The first group would be group A genes, which we hope to be a large subset of the total population, which would be genes that are expressed at a similar level in both the wild type and the BMPR2 knockout or BMPR2 mutant bones. We also expect to find genes in group B, which would be expressed at twofold or less lower in BMPR2 mutant mice. The third group would be group C genes, which would be expressed greater than or equal to twofold higher in BMPR2 mutant mice as compared to controls. This revealed a large amount of data. We observed somewhere in the neighborhood of 39,000 distinct hits on the RNA-seq data, the transcriptome profiling at 35-week-old mice, 35-week-old humeri. Among these 39,000, we noticed that there were reads for both exonic and intronic regions of the genome. We then filtered our data set to look at only those genes that we could be certain we were observing exons. Now this brought our filtered set down to approximately 25,600 genes that we were confident had exonic reads in the RNA-seq data. Filtering those then, we found that there were 922 genes which are differentially regulated in BMPR2 mutant mice as compared to control mice. About 350 were upregulated at greater than or equal to twofold, whereas 560 or so were downregulated greater than or equal to twofold in the BMPR2 mutant mice, leading us to 922 genes of interest in the study. Now, the challenge with RNA-seq data is weeding through the hay to find the needle. When we were evaluating these data, we examined it, in numerous different fashions. We chose 4 housekeeping controls that we felt were consistent between the control and the BMPR2 mutant bones. That was TATA binding protein, GAPDH, HPRT, and PPIB. This 922 number here that is represented are genes that are differentially regulated between control and BMPR2 mutant mice according to all 4 of those housekeeping controls. We have an additional set of genes that we aren't exactly sure what to do with yet, which are genes that are not detected in the control, or those genes which are not detected in BMPR2 mutant mice. And this is in the neighborhood of 350 or so for each genotype. We then looked through the list in the search for a smoking gun by evaluating the usual suspects. This didn't turn out to be as helpful as we had hoped, where this list of genes, which is related to osteoblast differentiation or osteoblast activity, did not reveal any significant changes between BMPR2 mutant mice and controlled littermates. SOST is the gene that expresses the sclerostin protein. This is the target of one of the anabolic therapies that was mentioned earlier in the slide. RUNX2 is a marker of the osteochondral progenitor lineage. SP7 is also called ostrix. This is a marker of osteoprogenitor cells. And then these latter genes here are related to osteoblast function: osteocalcin, leptin receptor, estrogen receptor alpha, alkaline phosphatase, and the vitamin D receptor. And again, none of these met our threshold criteria for being considered differentially expressed. There were, however, some interesting findings of genes that we weren't really expecting. Now, I should point out that the most strongly downregulated gene is the pseudogene GM5526, which has no known function, nor is there any way for us to follow up on this immediately. Likewise, the most highly upregulated gene was also a pseudogene, GM14925. There were, however, things which caught our eye that appear to be interesting for follow-up studies. One of these is a gene called PAC4, the protein of which interacts physically with BMPR2 and is phosphorylated by BMP signaling to promote cytoskeletal dynamics, and this has been demonstrated to be important for osteoblastogenesis. NDFIP1 is upregulated approximately 8.5-fold in BMPR2 mutant mice. NDFIP1 promotes the degradation of the TGF-beta receptor 1, which is a receptor in a family that in general controls catabolism of bones. It's a rather nuanced story, the role of TGF-beta signaling in the skeleton, but some of the effects of TGF-beta signaling are catabolic to the skeleton. Likewise, the TGF-beta, active TGF-beta pathway is downregulated by the product from a gene called TCEA1. TCEA1, the gene itself, is upregulated 6-fold in BMPR2 mutant mice. As was mentioned, this gene product reduces active TGF-beta signaling potency. One gene which we knew very little about before beginning the study is DYX1C1, which is upregulated about 6-fold in BMPR2 mutant mice. Knockout of DYX1C1 has been shown to cause axial skeletal patterning defects and also defects in left-right asymmetry. We find this interesting because not only is there an axial skeletal patterning defect, but breaking of left-right symmetry relates back to nodal signaling, and DYX1C1 mice have been demonstrated to have defects in primary cilia. This relates back to the TGF-beta and BMP pathway. The final gene I wanted to highlight today is CSN2, which is upregulated about 3.5-fold in the BMPR2 mutant mice. A study in 2012 indicated that CSN2 is a BMP pathway target gene, though this study was not performed in bone, and we're seeking to follow up those studies in our own hands. Now, in summary, I just want to leave you with these quick thoughts, which is that BMPR2 mutant mice display profoundly high bone mass in young adulthood. And here in this unpublished work, we demonstrated they also display high bone mass in late adulthood, so at least 55 weeks of age. BMPR2 mutant mice display elevated bone formation rate to at least 35 weeks of age. And we've now come up with a defined gene signature that is associated with elevated bone formation rate in BMPR2 mutant mice at 35 weeks of age. In follow-up studies, we are now repeating our transcriptome profiling at 55 weeks of age. Our idea here is to filter our results by moving to a later time point where P1 and P values are back to normal. And we hope to find that genes, we hope to filter through our genes at 35 weeks of age to identify those ones which are elevated only in the presence of increased bone formation rate or downregulated only in the presence of increased bone formation rate. That would be looking at the changes from 35 weeks to 55 weeks and finding overlap or differences there. We will then move on to validate a select set of genes by quantitative RT-PCR in the bones of these animals, and also using isolated osteoblasts that are expressing BMPR2 or have BMPR2 expression knocked down or knocked out. Then we're also looking at key signaling pathways in the bones of BMPR2 mutant mice. We're particularly interested in the interactions between the BMP pathway and the activator TGF-beta pathways, as our prior work demonstrated that that BMPR2 plays a crucial linchpin between these two pathways. And then finally, work in the lab is developing non-genetic strategies to target BMPR2 expression and/or function. And currently, we're evaluating a set of commercially available neutralizing antibodies for this purpose. I would like to acknowledge the people and funding that made this work possible. The first is my postdoctoral mentor, Dr. Vicki Rosen at Harvard School of Dental Medicine. And then the two medical students I mentioned to you earlier, Aaron Hudnall and Mike Eden, have been crucial for the success of these studies. Funding while I was a postdoc for this work originated from a postdoctoral research grant from the Harvard School of Dental Medicine. The work has been advanced by a series of intramural research grants from Marian University College of Osteopathic Medicine, and then, as was mentioned earlier, by a senior research grant from the Indiana Academy of Science. If you'd like additional information, please do shoot me an email. You can find contact information here at www.johnlowery.com/research, or look up the lab on Twitter using the hashtag LoweryLab, or my personal Twitter handle at JW_Lowery. We're always interested at developing new ideas, finding new collaborations. If you have a gene that you would like to sift through the transcriptome profiling and evaluate if it's changed in our mutants, please do let us know. Thank you.